Hello and welcome to this evening's UCSF Virtual Alumni Program, UCSF 3D Health Study, Data, Discovery, and Diversity. My name is Mario Peraza and I'm the Executive Director of Alumni Relations here at UCSF. Before we start tonight's program, I want to review a few housekeeping items to help with your experience. If you have any questions during the presentation, please use the Q&A box. Please keep your questions brief, and we did receive a number of questions before the registration, so we will get to those as well. But given the size of our audience and number of questions, our speakers may not be able to answer everything, but we hope you enjoy the conversation. Please let me introduce our speakers for the evening. Dr. Alexander Rejovic is a professor of pathology and obstetrics, gynecology, and reproductive sciences. He serves as the UCSF Chief Genomics Officer and is the medical director and chief of the Center for Genetic and Genomic Medicine that organizes, coordinates, and oversees clinical genetics and genomic services across the entire UCSF health system. He is also the director of the Genomic Medicine Initiative, a translational arm of the Institute of Human Genetics. And Sawana Biswas, who is a genetic counselor and researcher who cares for adults with concerns about genetic disorders. She evaluates patients for genetic conditions and provides counseling, including genetic testing and guidance. Our speakers are here tonight to discuss how whole genome sequencing helps scientists identify and respond to diseases much earlier using preventative therapies based on a deep understanding of disease biology. We are excited to have both of them here, and at this time, I'll turn the floor over to our speakers. Welcome, Dr. Rejovic and Sawana.
Thank you, Mario, and thank you for your team for inviting us. And also I would like to thank alumni who have decided to attend this session. It's very exciting for us to talk to you. I know that for some of you, it may be, if you're on the East Coast, it may be a bit later than here. So today, Sean and I will talk about our 3 Health initiative at UCSF and what does it mean for the precision medicine efforts at UCSF and UC as a whole. So just for introduction, this is me. I'm one of the medical geneticists and a chief genomics officer at UCSF. Shauna Biswas is a board certified genetic counselor. She is the scientific and clinical lead on the project. And there are a number of members on the team that have helped us in these efforts. And I would specifically like to acknowledge Jennifer Loano, who has helped in working with our participants and returning the results. Also, Dr. Keith Yamamoto, who is a big supporter of our efforts, as well as Benjamin Rubin, Courtney McFall, and others who are in the precision medicine group at UCSF. So today we will talk about genomics and specifically about precision genomic medicine. And I think I want everyone to keep in mind that health, our own health, is an adaptation to an ecological niche. This was well put together by Barton Childs. And it is important to understand that genomics, that is our architecture, really gives us a predisposition. And whether we develop a disease or not depends on a number of factors. And I like this spoken wheel kind of a presentation that shows that biology and genetic endowment do play a role in determining our health, but there are a number of other determinants that include health services, personal health, income, social status, education, physical environment, and so on, that together determines whether our genomic architecture will lead to disease or to a healthy being. And what I will be pitching today is that indeed the genome and how it actually interacts with the environment and how we can predict health is best done within a large academic medical centers with a lot of data sets that can help us determine the phenotypes. Now, the human genome, as we currently understand it, has about 20 to 25,000 genes. It's really, these only represent 1.5% of the genome, and we really only understand just about 1/5 of these 20,000 to 25,000 genes that are in the genome. So 99% of the genome, we do not understand its function, and we don't understand how it clinically affects our health. But we do know that at least 80% of the genome is somehow biochemically active, and hence our actual desire to look at the whole genomes. Now, Mendelian inheritance, which is pretty much defined, and this is what most medical geneticists study, is one gene, one disease. How can we identify one gene that is causing a particular manifestation, either cancer, heart disease, or structural defects? If we actually look at the Mendelian genetics, we now know that about 4,500 genes have some sort of a disease associated with it, or phenotype, meaning a structural, facial, or body feature, or a measurement, laboratory measurement that is affected by these genes. The most advanced clinical testing that is currently being offered in practice is whole exome sequencing, which is pretty much what is done in the laboratory. Is your genomic material is isolated, and then the DNA is broken up into pieces, and we're only capturing the portions of the DNA that code for proteins, while the non-coding regions are not sequenced, because we really know how to interpret only the coding portions of the genome. Whole genome sequencing is done very similarly, except we sequence both the coding and the non-coding region. But again, when it comes to clinical interpretation, even on the whole genome, it is confined to the coding regions of the genome and not to the non-coding regions of the genome. Now, with these incredible technologies that have become enabled because of the fruits of the Human Genome Project, which is one of those large science projects that has really paid for itself hundreds of fold by generating technologies where we can sequence genomes and so on, it has become clear that if an individual has their genome or exome sequenced, that you will cover genes that may be involved in cancer, such as colon cancer, breast cancer, or genes that involve heart disease or arrhythmias. And therefore, American College of Medical Genetics has come with a recommendation that if an individual, be it child or be it adult, that is getting sequenced because they have an indication for sequencing, that the clinician at the laboratory should return genes for which there is sufficient evidence that there is some actionability associated with them. In 2013, when this was first published, there was about 56 genes, then it decreased to 59 genes, and in 2021, it increased to 73 genes. And these are genes for which there is some sort of therapeutic intervention, management change, or surveillance that can be instituted, and for which there is some evidence that may lead to improved outcomes. And these genes, as I mentioned, they include cancer genes, heart genes, they include genes involved in neurological disorders, biochemical disorders, and so on. And if you were going to sequence the population at UCSF or in other places, about 2 to 4% of the individuals will actually have genetic variation that predisposes them to a certain disease. Now, that said, if we actually take larger number of genes, and for example, at UCSF, we have polled our experts who say that there is approximately 120 or so genes that they think there is actionability to do something about them. Then this 2 to 4% of the sample rises to almost 10% of the individuals will have something that actually that there is some actionable medical intervention. Now, at the same time, when these guidance were coming out, there also was an initiative, precision medicine initiative, to put resources towards trying to tailor medical care to an individual. And this concept of precision medicine, where you understand the biomarkers, genetics or others that make an individual distinct from others, and whether we can develop therapeutics that will be individualized. Of course, CAR T treatments and other treatments are there that are based on individual medicine. And so the concept was whether we actually should be doing also precision population medicine in genetics. Now, usual medical geneticists, they like to look at pedigrees. And pedigrees are very informative for many reasons. And usually based on the pedigree, a geneticist will determine whether there is a disease running in the family and whether there should be a genetic testing offered. However, many studies, especially when it comes to cancer genes, such as BRCA1, BRCA2, and others, we know that the pedigrees really only pick up about 30 to 40% of genetic pathology. So a number of individuals will not be sequenced just because the pedigree doesn't appear to be informative. Now, population-based precision medicine has already been practiced in the area of newborn screening. So newborn screening is an example where we, of course, screen every newborn for a suite of potentially serious disorders. And this population-based programs have shown significant benefit, are in place across the United States. And so the idea was to basically offer whole genome, to offer some sort of sequencing to populations, whether it's a panel of genes or whether it's whole exome sequencing. And a number of progressive health systems have actually begun to offer to their populations sequencing, both to have research purpose as well as the clinical purpose. And these are some of the systems that are actually shown here. So there is Geisinger MyCode, there is Intermountain Health, there is NIH, all of us that is trying to generate a large research repository. And at UCSF, we feel that the exome sequencing is not enough, that in order for us to really use the research capabilities and prowess of UCSF, we need to understand the non-coding region, and therefore the whole genome sequencing will be very important for the research efforts, in addition to helping deliver clinical results to patients. So let's actually look at what would it mean if you sequence 250,000 participants in our system. We know that approximately 2 to 4% of the individuals, if you only return these 59 genes, will have something that can be returned back to them and that is significant. And if we estimate how many participants will actually have pathogenic variants, we find that about 5 to 10,000 will have pathogenic variants. But it's also important to realize, because genetics is a familial thing, and genetic traits, of course, are inherited, that we also will identify approximately 15 to 30,000 relatives at risk. And so the total number of individuals that will be identified that will be at risk will be 20 to 40,000 individuals. If you look at the most prevalent conditions that one would identify using these particular screens, this includes familial hypercholesterolemia, hereditary breast and ovarian cancer syndrome, Lynch syndrome. Those are the 3 top conditions that will be seen. Now, in addition to this 59 or 125 genes, if you're at UCSF in the future that we will be returning, there are other things that can be returned to the individual. And that includes, for example, carrier screening. We all carry a certain traits that when inherited in a recessive fashion can cause severe disease in children and actually adults. And if you do whole genome sequencing in unselected group of individuals, and just focus on 700 or so actually genes out of 2,000 that are severe in terms of what they can cause. We find that almost 80% of the individuals will carry at least one positive result, which tells us that if we sequenced for all of the important genes, that all of us are carriers for a disease that we will transmit to our children, and hopefully they don't have a partner that carries the same gene change that can cause an affected child. Another whole suite of information that can be returned back to the individual is pharmacogenetics. Pharmacogenetics is a very active area. There are almost 140, more than 140 drugs now that have genetic data in their FDA-approved product labeling. And this, of course, is especially important for middle-aged individuals that are already starting to take medications, be statins, hypertensives, or other medications, and where pharmacogenetics can have a significant effect. So we are, we basically are working towards this learning healthcare system where we can use big data from the clinical research data as well as epigenomic data to basically improve our ability to predict and prevent. And the healthcare systems have already a lot of clinical data in them. You see, and as part of this, of course, initiative to add to the clinical data, the genomic data, which is lacking in many healthcare systems, including actually the UC system, we have then started the 3D Health Initiative. And the 3D stand for data, which is the whole genome sequencing data, the discovery, where we are partnering with other precision medicine efforts across UCSF to include genomic data in the analysis, as well as to increase diversity among our populations, because the equity and inclusion is a very important area in science and in medicine. And of course, how do we implement precision genomic medicine for all 10 years from now, that becomes incorporated into the practice, with really a goal that we can improve health and prevent disease with personalized care that hopefully will be practiced by primary care physicians, either healthcare providers and specialists. Now, within UC Health, as I said, there is very little genomic data, very little genetic data, but we are actually very wealthy with other clinical data, which also includes environmental exposures, other laboratory imaging, and social data sets, as well as the economic data sets. And UC Health as a whole has almost 15 million patients in it, so provides a very, very rich data set with all sorts of different clinical phenotypes onto which one can overlay the whole genome sequencing and find meaningful relationships. Across UCSF, there are a number of precision medicine programs. Some of them utilize purely what is currently available at electronic medical records, such as Bridge, to basically develop barcoding based on all sorts of clinical data, based on your age, based on your sodium, potassium, your blood counts, and all that, and make barcodes that associate with certain diseases, whether it's rheumatoid arthritis or whether it's multiple sclerosis or other diseases. And so they're trying to develop barcodes. But again, there is no genetic data in our system. SPOKE is actually another effort that is also trying to use our ability to utilize a lot of other data sets to provide the clinicians with more insight into the patient's health. Again, all based on clinical information and not on the genetic data. And then there is other efforts here that are listed Now, of course, to do precision genomic medicine, you require a significant team. Require genetic counselors. We are lucky to have Shauna Biswas with us. We require clinical lab directors, geneticists, clinical informaticians, primary care specialists input, especially on which genes are important for them and which are actionable, and of course, bioinformatics, bioethics, and the biobanking. And of course, bioethics is very important, especially in terms of issues of inclusion, diversity, issues regarding consent, and issues regarding data utilization. Now, we at UCSF have the first preventive genomics clinic in the country that was initiated about 3 years ago. This type of clinic now has been copied at Harvard and a few other places. We do actually see patients who want to get insight into their genomic information and to have either interpretation of commercial testing or whole genome testing that we offer. And it's really focused on assessment of risk in healthy individuals. So with that, we were funded by UCSF strategic funds to venture into this area of whole genome sequencing. And we focused here on adults, although we had a lot of pediatric colleagues that wanted us to also include children. And we wanted to include individuals who did not have known pre-existing genetic diagnosis. We targeted approximately 1,000 individuals across UCSF Health. Really our aims at 3D Health were to better understand and also develop what are the kind of electronic modalities that we need to recruit diverse group of patients, also how to engage that diverse group of patients, and we will talk about some of the stakeholders that are helping us with us, as well as investigate what are the best practices for us to consent, recruit, and deliver these results. So we did a lot of manual interaction with the individuals that participated in the study. And also we wanted to integrate genomic data into the electronic medical record and establish a repository of these data sets with the clinical data sets that can be used both by clinicians and researchers for future studies. So I'm going to turn this over to Shauna, who will talk to us about the study and what we returned and what have been our experiences to date.
Mm-hmm. Thank you, Alex. So currently in our study, in this pilot phase, we really wanted to understand what it will take for us to build this infrastructure internally and what the requirements of the future will be. So as we said, we are doing the genome, so that's a lot of information, but we are focusing on a subset of genes. So we are focusing on the ACMG59 gene list, which is convened by the American College of Medical Genetics, and it consists of genes that are medically actionable, and it's a well-established gene that is also a gene list that is also part of clinical testing, and generally the list consists of adult familial cancer conditions or cardiac conditions, and we expect about, as Alex mentioned, 2 to 4% of cases coming back with a change in one of these genes which are actionable. And as part of this study in the first phase, we also wanted to return ancestry, and we will have a comment on that a little later. But as regards to our process, it is in this first phase, it's very, very manual, very labor intensive. Our patients contact us, or we have different modalities to contact our patients through our electronic health record, MyChart system, or even we utilize some social media tools to reach out to our participants, people who are interested. They send us an email or a call or a website request. Then we go through the whole process of consenting. We send the saliva kits to our participants' home address, and that goes back to the lab where we do the whole genome sequencing. So this is a clinical grade 40x genome, so a very good coverage. And then we create a clinical report. So this is in a CLIA environment that tells us, among those 59 genes, if any pathogenic or likely pathogenic mutation was detected. And then a clinician will return those results back to each participant and also share this with the primary care doctor and put it back in the chart, which does close the loop and also allows for future research on the data and continual input of clinical data set. Next slide, please. Okay, so just a quick timeline of that. It generally takes about 6 to 9 months for us to get the results back, and then the research will continue for several years. So when it comes to data and leftover samples, we have samples that we are collecting from clinical testing, or they are banked in our biobanks. So we have also, language in our protocol where we can collaborate and also retrieve those leftover samples. But currently from the study, the data will be de-identified. So all the raw genomic files will be linked with the electronic health data. And we will also include, if any, biobank data that is linked to those files, and combined, all of that will stay in one place so that you can use your internal my access portal and then access this information in a combined manner, so that you can say, okay, show me, you can search by genes, you can search by phenotype of interest, and then you can look into those genomes and find out a gene of interest or a variant of interest. We also are trying to put together a protocol or a framework where there will be oversight on how we will use the data, who will use the data, and some guidelines as to sharing the data. So that is also being part of this project. In terms of recruitment, we did launch a year back. So this was during COVID. And as you guys know, there were some challenges and we had to adapt. So initial plans were changed, and we had some ups and downs. We utilized our electronic tools. We also utilized our PR and social media to drive. And also we figured out maybe identify champions within the primary care clinic. So we worked very closely with providers in our DGIM clinic and dental clinic, and we are trying to actively recruit through that model. But it is very resource dependent and labor intensive too. So currently we have 500 plus participants, and we continue to enroll in future too. In terms of our distribution of our cohort, the age of our cohort is pretty well distributed. We do have a good mix of elderly and young participants. So people are really interested in knowing their health. We also have a slightly higher contribution of females over males. Next slide, please. And in terms of the results from these genomes, so we have completed about 472, and out of that, 17 has come back positive, which means that there is a change in one of those 59 genes recommended by ACMG. So that equates out to 3.6%. And on the right, you see a composition of the ancestry that we can pull from a genome. genome. But there are current some limitations because in terms of reference data, we can only have or refer back to 1,000 genomes, which are not enough, and there are some limitations, and we are actively working on finding a good solution. So in terms of the results, the results do, once we have a positive ACMG59 test result, which has an impact on that particular person's health, so Alex and I, we both see the patient, we go through counseling, we give the patient information as to what this means for the patient and for their family members, and we also provide recommendations and referrals to appropriate departments. So it does go back, even though this is a research study, it actually is also a medium where it connects directly with clinical care, and there is an impact in patients' health and patients' chart with the documentation of what that finding is, so that immediate action can be taken on this result. So just a quick, overview or a few cases here out of those 17. We have participant 1, who is a 73-year-old female with a history of hypertension and coronary heart disease. And there was some family history of cardiac arrhythmias, and she was found to have a KCN1Q likely pathogenic mutation, which is associated with long QT. So the follow-up was recommended with a cardiologist, and she did get implanted with a defibrillator. The information was critical for her brothers. Both appreciated the information and also did have genetic testing locally, and one of them were found to have the same mutation. Participant 2 is a 38-year-old male in good health with no family history of cancer, was found to have a BRCA1 mutation, and is now being followed by our hereditary cancer program, and undergoing some at-risk individual testing. Two of his siblings, one of them is a female, is undergoing currently testing. So that has an impact on other family members. Participant number 3 is a 61-year-old female with a past history of breast cancer, but also had this history of shortness of breath during exercise and 1 episode of fainting. And she was found to have a mutation in SCN5A, which is associated with Brugada syndrome. So this is also another outcome that she did not expect. She was referred back to cardiovascular clinic, and she also shared this information with 3 of her siblings, and they are interested in testing. So that kind of gives you a flavor of how our testing, our preventive testing, has made an impact in some of our participants. We also try to jot down the motivations for different participants to join in our study, or even go through sequencing. And 2 big themes that came through is, one is in the altruism. They really want to promote science or contribute to science or research. They also have a desire to learn more about their own health and what they need to do differently. And some other themes were people were seeking information just for their family members. And most people also said that they trust UCSF and they want to give back to UCSF. Some people also wanted a copy of their own data because they understand the significance of that, or they are in this field and interested in it. We also did some patient surveys, and some of the preliminary results do show that 99% of our participants gave permission to include this result in the electronic health record. So that's great confidence, showing that they are okay being shared and they understand that this might have some implications on their health. 78% discussed the results with a physician or a family member member, and 6% reported an additional appointment with a healthcare provider. And many participants, at least 93%, did feel good about helping the medical community, and 80% felt this is an extremely important or very important health effort. So moving into slightly in a different gear, the last D, diversity. So we do have an aim where we want to focus our efforts on what it will take for us to increase participation from diverse participants, because genetics or genetic data on certain ethnic backgrounds is lacking. Like predominantly our data set is Caucasian based, and so whenever we find these variants, and it's most of the times it's uncertain because we don't have another data set to compare it with. So it's becoming more and more important that we need to sequence more participants from different backgrounds, and we want to identify ways to do that. So currently, due to some limitations from COVID and the mediums that we can use, our demographics is 76% Caucasian, Asian is 17%, Hispanic is 3%, African American is 2%, and the remaining is mixed or other. Now, the numbers that are in orange are the ones that are represented from the UCSF patient population. So we, there's a big difference between 76% Caucasian versus 46%. So we have a lot of work to be done, and we need to up our numbers, from our current numbers. And for that, we have partnered with our collaborators in SPEAR. So that's a special populations and health equity in research and education group. And their main aim is to really promote diversity amongst research participants through community engagement. And for the 3D health study, they helped us serve as an advisory body, and they really helped us understand what are the barriers here. So in terms of the team, we have a very robust team, and we have representation from different communities. Joyce Chen is a representative from the Chinese community. Angela Costello is from the Latinx community. Monique Lasser is from the Rafiki coalition. So we do have many more names, but we do interact with them pretty often, and they consult with us. In terms of where, how the SPEAR committee has helped us, they has helped us to address what are the different trust issues, how we should adjust our materials, how we should incentivize our participants, how to expand our recruitment sites, and what should be the modes of expansion. We also tried to conduct, town hall meetings with our community leaders and representatives, so we are in, how do we increase those numbers, and they are continuously advising us throughout the process as to how to scale this up and also ensure equity and inclusivity.
Thank you, Shauna. I think, as you can see, Shauna has done yeoman's work here in returning the results to almost 500 actually individuals, and we still have approximately another 500 to recruit and sequence. And so as part of our future efforts, and as part of our highly manually intensive experience, we are basically working to develop digital tools that will allow us to capture all individuals that go through the UCSF health system and that want to participate in these endeavors. Now, although we have initially focused on UCSF Health, and I know there are some individuals outside of the Bay Area who are not UCSF Health participants, and also alumni, that we hope to expand this effort to them also, including our affiliates, as well as public hospitals. Now, we would like to scale up our efforts to sequence approximately 100,000 individuals over the next 5 years and expand what we return back to our individuals, including pharmacogenomics, other actionable genes. As I mentioned, UCSF faculty feels that there is 120 genes, which almost 10% of the population will have something that is actionable, as well as polygenic risk scores. Polygenic risk scores are entering medical practice. They are not Mendelian genetics based variants. They are variants derived from genome-wide association studies and show quite a promise in helping refine and improve our efforts to predict disease. We also are working on integrating how to best integrate this into the electronic medical record by developing best practice alerts that will alert the primary care physicians who are busy doing a lot of different things, and alert them in terms of what does this mean for their patients. Now, another important thing is to keep regular communication with the participants. And this is done through reanalysis of data. The data can be reanalyzed anytime. In most of our experience, yearly reanalysis catches significant advances and can make differences, especially in patients where we didn't really find anything. And this is the way that we hopefully keep connected to the individuals. There also is discussion whether we should be including pediatric populations. And I do think that this is the next frontier. And basically engage many of our research efforts to integrate genomic data into other precision medicine efforts, because as I said, there is, we are a system that is very rich in clinical data, but very poor when it comes to genomic and omics data. And of course, we are saving extra tubes of blood for additional omics in the future. And of course, this will all be as part of the research, and one of the tracking is to really look at the clinical outcomes and utility of our research efforts. Now, some of the requirements for scaling are basically that we are working on e-consents that will reach all of our populations in various languages. As Shauna said, we have had, there is a discrepancy between what is UCSF Health demographics overall and what we have recruited, but partially because our pilot was limited to English-speaking individuals, which can be a barrier, of course, to a number of patients that we serve. So we need to involve translation in many different languages. We also need to have ways to communicate this that is not so labor-intensive. So developing portals and way to communicate digitally, where patients, of course, can always reach a genetic counselor or a geneticist for help, but that most of the negative results do not require interaction with a physician. And then, as Shauna mentioned, the ability to store this information, link it to the electronic medical record and all the other data sets that are important in order to find new discoveries and new phenotypes. And the goal really that in 10 years, we will be practicing precision medicine, genomic medicine for all, and that hopefully we'll be able to integrate all of these determinants of health together with the genomics to be in a better position to prevent and predict outcomes. And so this is the website for our study. You can learn more about going there and reading about it. So thank you all for your attention. And now we can try to, we have, I think, 15 minutes where we can try to get to some of the questions that have been asked by the webinar. So the first question is, how do you envision the interface between the patient and the UCSF Medical Center in 2035, when clinically useful genetic and genomic data about the patient will be available at his yearly preventive interaction with the system and its practitioners? So as I mentioned actually at the end, I think the genome, as well as many other things, it really provide hopefully a umbilical cord to our participants, to our patients, so that we will be able to, on a yearly basis, review genetic information, review what is new and what actually is actionable, what is clinically meaningful, and provide that patient with results. If these results are meaningful and have findings that require further follow-up, then we can direct them to the appropriate specialists at UCSF for follow-up, maintenance, surveillance. And so this is how we envision the interaction between the patient and their genome at UCSF. Now, please address the harms and unintended consequences, if any, can be imagined for whole genome sequencing and disease prediction models. I think this actually is a great question, because with every biological or discovery good, there also is a potentially an adverse event and consequence. As I told you, we don't understand what 99% of the genome still does. And I think some of the harms and unintended consequences is misinterpretation or overinterpretation of the data. Because the genome is vast, not well understood, there is a lot of variance of unknown significance. And for foreseeable future, the interpretation and ultimately the result will require presence of a geneticist or counselor, genetic counselors, or other healthcare providers that are versed in the arts of genetics to be able to put the genetic information together with all the other information in order to make a meaningful recommendation. Because as I said, the genome by itself, without all the other health determinants, is very difficult to interpret. And so I think we need to always keep in mind that that we don't overinterpret the data that we have in hand. This actually is a question for Shauna. And how will patients receive messaging that still encourages lifestyle measures to prevent disease rather than targeted treatments?
Yeah, that's a great question. So right now we are in the process of identifying the best method to communicate these results to our participants. So we are in this pilot phase. We have started returning results. In some cases, it has made an impact, as we had highlighted in some of the case presentations. If you have a BRCA1 mutation, then there are some changes that you can implement, or if you have a certain cardiac finding, then there are some dietary changes or other lifestyle changes that you can implement. So we do communicate those in a very simplified manner, but also as we proceed into the next phase, there are so many other genes and so many other domains that we need to look into. So that will be an active part of research, and how to communicate is something that we will be actively working on.
Yeah. Thank you, Shauna. Another question is about, I'm especially interested in an update on how genomes and phenotypes interact. What are we learning beyond dominant and recessive about the mechanisms that affect phenotypes or manifestations of the genome, and how do they change the actual genome? And so I think that this is, of course, a cutting edge in terms of genetics, moving away from what we call one gene, one disease, and multiple genes and how they affect the disease, and how multiple gene actually interactions result in some individuals manifesting a disease and other individuals not manifesting a disease. And so the polygenic risk scores have become the new cutting edge. It's still in the research realm, but there are some conditions such as breast cancer, prostate cancer, where some health systems have started to use polygenic risk scores to see who is at risk. And what researchers have found is that the polygenic risk scores can have as strong of a prediction just as monogenic diseases. And so this is pretty powerful. But as mentioned previously, unfortunately, because most of the genetic information so far has been derived from Caucasian populations, the usefulness of polygenic risk scores tends to be significantly better in Caucasian populations because we have much more data than it does in other geographic groups, racial and ethnic groups. And so there is a significant effort to close these gaps. There is actually an African Genome Project. There are genome projects across many countries that are helping us better understand the diversity. And that's why the whole geographic ancestry is important, because it is used to derive these polygenic risk scores. And it's something that will happen in the near future. And we are working with internally with a number of UCSF researchers that have expertise in ancestry as well as in polygenic risk scores to start implementing this in actually our clinics. There is another question that asks about how will the study ensure there is representation of diverse segments of the population. Shauna, you want to answer that question?
Yes, so we have in our talk highlighted how we are collaborating with the SPEAR group, and also some of our participants who are from diverse backgrounds are also motivated in helping us, either like spreading the word through their community locally, or even through the SPEAR team, trying to address some of those barriers and trust issues, and how to share and how to talk about genetics to different groups. That is also something we are learning and adjusting.
There is another question about, can you clarify the exclusion criterion that a participant does not have a previous genetic diagnosis?
Yeah, so that specifically means that you did not have that specifically means that in the past you did not have a genetic test with a finding. So if you had gone through a genetic test or a panel or sequencing and it came back with a change or a variant, then we will not be able to recruit you. However, sometimes certain diagnoses are also clinical diagnosis. Like if you have a clinical diagnosis of EDS, we will not be able to recruit you in that instance.
There's actually another question that it's relatively specific, but I do want to address it because it shows sort of the power of potential work against UC system. There is a question about, are there genetic markers that tell us what type of interferon cascade a patient has? This could lead to a better understanding of how the immune system reacts to an infection such as COVID. So Shauna and I, during the COVID crisis, we did actually start a UC-wide COVID consortium to sequence genomes of individuals that were hospitalized with COVID. And we started a consortium that included all of the UC health systems as well as non-health UC institutions that were interested in this research. And we, as a group, have been able to sequence over 1,000 genomes and have participated actually with other consortia that have propped up with efforts to better understand why certain individuals will develop COVID and other individuals do not actually end up hospitalized with COVID. And so these efforts are still undergoing, but it also shows the power of the UC to get together, and also the power of sharing de-identified data to help us better understand the host responses. If, for example, we had everyone's genome sequenced in the system, it would have been very easy to pull out all the individuals that developed COVID, and potentially it could have taken much faster to find a host response that either protects or actually makes you worse. But because there is so little genetic information available on individuals in our system, that's not something that we have been able to do. But we are doing it now, and hopefully there will be a lot of insight from ours as well as efforts of others. Now, there is a question about is there a possibility of law enforcement using this data to match previously unidentified genetic data to criminals or their families, etc. So genetic data, as you very well known, is very popular in ancestry, determining where geographically you're from. And of course, the law enforcement uses it to find if individuals are related to a particular person of interest. And yes, this data, of course, can be used to identify whether you are related to someone or not. So, of course, for that to happen, the data sets have to be in a public database and available for others to peruse and compare, which these data sets are not going in public databases. Now, that said, yes, you can use genetic information to determine if you are related to someone. And of course, you need access to other individuals' data sets and their genetic information. But beyond that, there is very little that an individual that hacks your genome can do with the data. There is very little in it that is going to be interpreted for, I guess, nefarious means, if I wanted to say that. But that's why we take data privacy and very seriously in the area of genetics, and we have there is the whole bioethics team, and there is actually a lot of even research among bioethicists about how the data should be shared, what's the best way to share it. But it's important to understand that in terms of research, N is equal 1 is not enough. To find new discoveries and to find new associations, one needs larger numbers. And so participation in data sharing is actually very important. And de-identified data sharing is something that has become an accepted practice in medical research, and has led to many, many discoveries up until now, and will lead to many more discoveries in the future. With that, I think that we have probably addressed most of the questions, Mario.
Yeah, thank you guys so much for being here this evening and giving us such a great insight into the work you guys are doing with whole genome sequencing. So with that, I want to thank Alex and Sawana for the work and everything you're doing for UCSF and taking the time to chat with us this evening. For all of our alums who are able to join us and for your outstanding questions, thank you. We'll end the evening here. I hope everyone has a great night, and we'll see you at the next alumni event.